off class last week talking about Mr. Mendel and his peas. So even though in the 1800s or so, Mr. Mendel did his research, what we've come to find out is a lot of what he established as far as research goes still holds true today. Now, alleles, what were those? Right. So alleles is a section of your genetic material that codes for a certain thing. Now remember, your genetic material codes for what? What is it? What's it for? Your genetic material, your DNA. What's it for?
Everything about you.
Like what?
Like hair color, eye color.
Which is all what? Which is all made of what? I'm getting there. We're getting there. You're right. You're all right. Proteins. That's what I'm looking for, right? So alleles are different sections of the genetic material that code for a certain protein. So alternative versions of genes are called alleles. Different sections of the genetic material that code for a specific protein, for eye color and hair color and all the things you guys were saying. So you're right. So for each characteristic, for each protein, you have two alleles. So when we talk about eye color and we talk about hair color, I hate to throw those out there because some of them actually are more than one allele or more than two alleles. But you get one pair of information from each parent for your pair of information for whatever protein we happen to be talking about. So for every single protein that your genetic material codes for, in your whole entire body, you have two copies of that. Or two what? You have two pairs or two alleles. Yes? So if those two alleles have the same information, because you don't necessarily have the same information from both parents. You have the alleles to code for all the things that you need to make for you as a human being. But they don't have to be the same. Exactly. One can be blue-eyed, one can be brown-eyed. But they're both for what? Eye color. See what I mean? If you have the same information from both parents, it's said to be homozygous. So your information that you have for, let's stick with eye color, if it's the same from both parents, brown eyes, you're homozygous. Both alleles are coding for the same protein. If you get two different bits of information from your parents, one brown eye, one blue-eyed, you're said to be what? Heterozygous. So the information, the protein that's made, even though they're both for eye color, they're different. Heterozygous. So you need to understand those two terms. So if two alleles of an inherited pair are different, sometimes one of them kind of masks the other. So if I draw this line here on the board, and then I draw this line over it, which one do you see? You see the blue one, don't you? Is there both there? Well, if I see one, even though both are there and they're different, that one is said to be dominant. If I see that. There's two of them. I'm going to see that if there's two of them. Yes? If I had another color to go over it, unfortunately with these markers, that's not going to work. But if I drew like yellow, for example, and then another color, you'd have to have two yellows to see yellow. Yes? Otherwise it'd be masked by whatever the other color was. If that's the case, what do we call that? That's recessive. Okay? So if they're only noticeable if I have two copies of them, those are recessive genes. If they get washed away by another gene called a dominant gene, do you see what I'm saying? Doesn't mean you don't have them both, just like it doesn't mean I didn't draw both those lines on the board, but the only one you see is the dominant allele. So dominant alleles and recessive alleles are also what we're going to talk about in genetics as well. So gametes are only going to carry one copy, right? Half the genetic information, one copy of each allele. Two alleles for a character are going to separate separately in those gametes. When we talked about meiosis, we talked about the separation of the pairs. Remember that? This is the law of segregation. So each of the gametes is going to have at least one copy, or one copy, of the information associated with each of the proteins that organism's going to make. And the reproductive cells are only going to have one copy of that. They'll segregate, the law of segregation. All right, do Mendel's hypotheses account for the 3-to-1 ratio he observed in the F2 generation? We're talking about, remember the purple plants? Okay. Do those things account for that? Remember, the offspring are going to have two copies. And I'm referring to, if you want to have a peek back, it's on page 149 in your textbook. Figure 9.6. We see that little diagram with the Punnett square that gives us the probability of offspring. And when we talk about the F2 generation, in this case it was the offspring of two purple flowers. But those purple flowers were what? Heterozygous. Very good. And that meant what? So they were this. Yes? And when the gametes got made, what do we see? I'm not going to change my markers because it's going to be a pain in the keister. Does everybody know what I'm doing? Okay. So this guy's white, and all the rest are purple. So does all that stuff we just talked about account for that? Good. So a Punnett square highlights 4 possible combinations of the gametes and 4 possible offspring of that F2 generation. And that's for the monohybrid cross. Sometimes we can talk about more than one trait that happen to segregate in different ways. And that's when things start to get confusing when it comes to the Punnett square. Okay, you saw some of them if you read your textbook, yes? It's nice and easy when we have one trait, monohybrid. So mono means what? One, exactly. So, we see our purple flowers, the original true breeder parent. That means what? When I say true breeders, what's that mean? They're purebred, exactly. They have 2 copies of the what? The same alleles. So we took our purple flower that had 2 purple alleles, and we crossed it with our white flower that had 2 white alleles. Now, because white's a recessive trait, that one's easy, right? If you're white, you have to have what? 2 whites to be white. It's a recessive trait. But purple, we have no idea what you are, right? You could be homozygous, like that guy, purple, or you could be what? Big P, little p, or W, whatever we want to call it. You could be heterozygous. I wouldn't know the difference. When we cross those 2, all the flowers are going to be purple. So what's one of the ways I could figure out whether or not I had a heterozygous or a homozygous dominant parent? Breed them with what? Yeah, with a what? A recessive individual. If all the flowers come out purple, chances are what? I'm a homozygous dominant guy. But if some of my flowers come out white, what's that mean? I'm heterozygous, okay? So we see that in the F2 generation when we cross 2 heterozygous plants, we see that trait coming back. Because what would our Punnett square look like if I did this? Yes? See my ratios? So they wouldn't all be purple. Half of them would be what? Half of them would be white. And that's how I would know I had a heterozygous dominant individual. Make sense? Okay. And it's easier with things that have a lot of offspring. It's hard when you only have 1 offspring. Because what are the chances that I'm going to get a purple flower in this match? 50-50. If I'm a human and I only have 1 offspring, and say I have 3 kids, all of them could be purple, and I still wouldn't even know that I was heterozygous, right? Because every single time I have an offspring, there's a 50-50 chance it's going to be purple in this case. I'll give you an example. I have brown eyes. My father has brown eyes. My mother has brown eyes. Both of their offspring, my sister and myself, have brown eyes. I married a blue-eyed gentleman. My first son has brown eyes. My second one, daughter. Guess what color her eyes? Her eyes are blue. Now, she could have had brown eyes. I never would have known. I have what? I have a blue-eyed gene. Guess where I got it from? My grandparents. My parents, both brown eyes. Exactly, but we didn't see it. Do you see what I mean? We didn't see it until it skipped a generation. Even though it was there, it was sort of masked. You couldn't see it. So I'm heterozygous blue-eyed, and the reason I know that is because I have a blue-eyed child. And then when I look back, and that's why you can see, and I think they did some of it in this chapter, genealogy. You look at the traits of your grandparents. My grandfather on my mother's side has blue eyes. There's where my blue-eyed gene came from. So, they distinguish between an organism's appearance, what they look like, because I have brown eyes, yes? What I look like is what you see. What do we call that? That's your phenotype. An organism's genetic makeup is called their what? Genotype. So what's my genotype for eye color? I'm heterozygous, brown-eyed. I have a brown-eyed gene and I have a blue-eyed gene. But what's my phenotype? I have brown eyes. A gene loci, or locus, is a specific location on a gene along a chromosome. So on whatever chromosome I happen to be mapping out, a specific portion of that chromosome, and I can't tell you off the top of my head because I don't know, is for eye color. That's called a locus, or a loci. Homologous chromosomes have alleles, different versions of a gene at the same loci, or locus. Again, some people are heterozygous and have the same one, and some people, homozygous and have the same one. Some people are heterozygous and have different ones. So we see this person, or whatever this gene happens to belong to, at a specific portion on the map, they see A, whatever A happens to be, they're homozygous for that A. Whatever P happens to be on that same pair of genes, this particular person is homozygous for P. And then for B, whatever that trait happens to be, this person is what? It's heterozygous. If B, capital B, the dominant portion, is something that will mask the recessive portion, what phenotypically are we going to see in this person? Yeah, that dominant B, whatever it happens to be. Make sense? Here's where we start to get a little confused. It's the same principle, except we just have a lot more stuff to keep track of. Mendel's law of independent assortment, that these guys get handed off independently. Some of them we see when we talk about genetics now, we see they're kind of linked together. But remember that whole crossing over deal that we talked about when we talked about meiosis? We talked about sometimes shuffling of genetic material. And this is kind of what we see in Mendel's law of independent assortment. So if we look at two traits, a dihybrid cross, the mating of parents with differing in two characteristics, what would the result of a dihybrid cross? Two hypotheses are possible. So whatever the trait is that we happen to be looking at paired together, they're either going to be dependent on the other as they're passed on, or they're going to be passed on separately. So how do we figure out which is which? Well, again, Mendel was into the P's, right? So he looked at a couple of different traits to see if they sorted independently or they were linked together and sort together. So he looked at seed shape and he looked at seed color. And we're talking about the seeds inside a pea pod. So big S would be smooth and little s would be wrinkled. Which one do you think is dominant? Smooth, exactly. Seed color, big Y would be yellow and little y would be green. So here we have heterozygous seeds. Dominant alleles for yellow peas, dominant alleles for smooth peas, and recessive alleles for green peas, and recessive alleles for wrinkled peas. So what do these parents look like? Yeah, they're smooth and they're yellow. But they're what? They're heterozygous. So when these alleles separate, they can only give one copy to their gametes, yes? They have two copies, though. Now we're starting to get a little confused, or confusing. But basically, we just have to keep those alleles straight. There's 4 possible gametes that can be produced by these heterozygous parents. And I'll show you on the board in a minute. So now we have a whole bunch of different offspring because we're talking about 2 traits that sort independently. And we get a ratio of 9 to 3 to 3 to 1. Again, we're talking about a heterozygous trait, dihybrid cross that sorts independently. What I want you to do is keep all those terms straight in your head. Understand what each of the things means, right? So what's a dihybrid cross? We're talking 2 traits. They sort independently. So remember, I can only give one copy to my gametes. But since they sort independently, I can have a whole bunch of different combinations. So let me show you what I mean. Let's use blue. So if I have smooth and I have yellow times a smooth and yellow parent. Why did I put those little things there? Now, he said it was a hybrid cross. So what's that mean? It means the other one's going to be what? Yeah, different. I'm going to put a little cross through the S so you can see the difference, and a cross through the Y. They sort independently. So when I talk about my gametes, each gamete that comes from that parent can only have one copy of S and one copy of Y. But because the 2 copies are different, there's a whole bunch of different possibilities for my gametes. So let's make the gametes and try to keep them straight without confusing them. So I can only give you one S and one Y. So what's the first gamete going to look like? Big S, big Y. Yep. That's 1. Keep the big S. Use the second Y. Big S, little y. That's 2. Yep. Now let's use this S. Little s, big Y. Yep. And little s, little y. You got it. You see where the 4 gametes came from? So those are all the possible combinations because they sort independently. Smooth and color and seed shape sort independently. So it's a crapshoot what I give you. And these are all the 4 possible gametes that I could have in this particular mating. from these particular parents. Yes? They're both the same, so we put their gametes across the top and across the side, male, female. And then we put them together. And we see all the different possibilities for the kids. Now, for the dominant traits, they tend to be that higher number. So how many of these guys are smooth and yellow? Yes. Even though some of them are homozygous for both traits, or homozygous for one and heterozygous for another, it doesn't matter. Dominant, you're going to see it. Yes? So these are phenotypes. 9 of them are going to be smooth and yellow. What's the other combination? Smooth and green, wrinkled and yellow. So 1 dominant and 1 recessive. Yes? And then one of the guys is going to possess what? Both recessive traits. So the 9 to 3 to 3 to 1 ratio is typically always seen with a dihybrid cross, 2 traits that talk about traits that are dominant or recessive. Yes? If they sort what? Independently. Whew. Yes? Yes? Come on. Here's another one, or another way to look at it. So if it was dependent assortment, that means they sorted together. This would be the outcome. Is that the outcome? Yeah, no. This is the outcome. So these are the actual results. So do we have dependent assortment or independent assortment with these particular traits? Independent assortment. Okay? And do you see why? Because we're going to get all kinds of combinations, which is what we actually see. These predicted results, if we were to guess it was dependent assortment, we don't see in the offspring. Okay? So again, Mendel figured this out way, way back when. And we know it holds true today. So Mendel's dihybrid cross supported the hypothesis that each pair of alleles is going to separate independently. The inheritance of one character has no effect on the inheritance of another character. And that's the law of independent assortment. Again, that's not for everything, but in these particular cases it was. Independent assortment is also seen in 2 heredity characters in Labrador retrievers. Cute little puppies. Now, coat color versus vision. So we have a dog, black coat, normal vision. We have a dog, black coat, that has what? Abnormal vision. They're blind. If they assorted dependently, we would expect to see everything with a black coat being blind. Yes? But what we do see is not quite that. Yes? In this case, again, we don't know what the other gene is for this dog, but if we cross it with a blind animal, we know that blindness is a recessive trait. They have to carry both alleles in order to be blind. Here are some of the phenotypes for the Labrador retrievers. And this is what we get when we cross. So, mating of double heterozygotes. What's that mean? You're heterozygous for both traits. You carry both alleles. When we cross the two, this would be our ratio, which is what? 9, 3, 3, 1. A chocolate lab is what? Yeah, it's recessive to black. And in order to be a chocolate lab that has impaired vision, is blind, what do we have to carry? What would our genotype be? Yep. So we'd have to carry both recessive in both traits. So this dihybrid cross, does that prove law of independent assortment? Yes. So in order to see what your dominant breeder has for genes, you have to do something called a test cross. So just like in the plants, in order for me to see whether that purple plant was a heterozygous purple plant or a homozygous purple plant, what did I have to do? I had to breed it with what? A recessive. If I bred it with a purple plant, yeah, it wouldn't tell me anything because everybody would be what? Everybody would be purple. So we do something called a test cross, and that's a mating between an individual that has that dominant phenotype, and basically we don't know what the other one is, and we cross it with somebody that's only giving those recessive genes. If your offspring have what? If all the offspring are purple here, what's that telling me? Chances are, and even then you can't always be 100%, that this is a heterozygous individual, or excuse me, homozygous individual for purple. But if some of your plants are white, what does that tell me? This is a heterozygous individual. So that's a test cross. So we could do a test cross with the two dogs. And if we had any of the dogs coming out chocolate and blind, we would know that that dog was what? Heterozygous for the trait. So two possible genotypes for the black dog, color-wise, are going to be big B, little b, or big B, or little b, right? Big B, big B, or little b. Yeah, for the offspring, just for coat. So here's the two possible genotypes that we could see for the offspring of that black dog. So if that black dog was heterozygous, what are we going to see? Coat color. If we cross it, test cross it with a chocolate lab. Nope. If it's heterozygous and we test cross it with a chocolate lab, some of the offspring are going to be what? Chocolate. Again, that's not necessarily the case because it's a 50-50 shot every time, right? If that dog was homozygous dominant and we crossed it with the chocolate lab, all of its offspring are going to be what? Black. If I took that guy and crossed it with a chocolate lab, then I might get some chocolate labs. So we can actually, we've done this for years, breed for specific traits. like chocolate labs. Why is there so many more chocolate labs now? Yeah, because they're desirable. People like them. So if I get a chocolate lab out of a litter, what am I going to do? Grab him out and do what? Breed him. So Mendel's strong background in mathematics possessed him to use all of these different mathematical rules. Rule of multiplication states that the probability of a compound event is the product of a separate probability of independent assortment. What the heck does that mean? This is where Mendel's statistical background came out. Who's taken statistics? Okay. So you know exactly what we're talking about, right? Yep.
I did a long time ago.
I know. But hopefully this makes it simpler to see. So if I take a coin and I flip the coin, half the time I'm going to get what? Heads, and half the time I'm going to get tails. There's only two sides, right? There's only two possibilities. If I flip it twice, is that going to be good for me to figure out the probability? No. You have to flip it what? A whole bunch of times. But pretend we flipped it a whole bunch of times. And we saw that, and again, we're calling these eggs, but basically they're coins, right? So if I have two alleles, if I'm a heterozygous individual, or I'm a coin with two sides, do you see where we're going with this? The probability that I'm going to flip one side or the other is half. This is the law of multiplication. Same thing for that other coin, heads and tails. So we have a quarter and a Susan B. Anthony dollar. Both two sides, heterozygous, yes? So every time I flip that coin and get heads, the probability of me getting heads would be 1/2 times 1/2, which would be what? 1/4. The probability of me getting a big B, a head, and a tail would be 1/4, which is what? 1/2 times 1/2. You see where we're going with this? So that's the law of multiplications. Little b, big B, same thing. 1/2 times 1/2 is 1/4. And little b, 1/2 times 1/2 would be 1/4. So what, instead of doing all the math out, what does this wonderful Punnett square give me the answer right away? It gives me the probabilities right away, right? Without having to do any math. If 3 of these guys are what I want, or the same thing, then the probability would be 3/4, which would be 1/2 times 1/2 times 1/2 times 1/2 times 1/2 times 1/2. Yes? But it's easier when you see it this way, isn't it?
No, I like it better the other way.
Well, this just adds the math to it, because he was a mathematician. So we got to give Mendel his due. He did a lot of work for us. So pedigrees, what our families look like, all of our different traits. Mendel's principles apply to the inheritance of human traits. Because we have so many different traits, sometimes it's difficult to pull them out to see, to look at our pedigrees. But this is when we look at our family tree. So, he pulled out some different traits, things like freckles. Who's got freckles? Yeah, they're there.
Now, but I do.
Yeah, she got freckles?
I have a daughter with one of my 3 daughters has freckles.
Yeah. And what's freckles? One of your 3 has freckles. Who else? Does you or your husband have freckles? Okay. So you must be what? And he must be what? He must be what? You must be. You're recessive, right?
Recessive.
What's he?
He's dominant.
but he's heterozygous. Yes? You see where we're going? Because if what did you say? One of your kids has freckles and two of them don't? Well, two of them are recessive. So what's that mean? They're homozygous recessive. So this is your husband. I'm going to put big F and little f. And this is you. Yes? Make sense? Because you don't have freckles. You're what? So there's your test cross. You did it right in your family. Yes? And that's how I know what hit. If all your kids had freckles, would we know what your husband was? No, because you only had how many kids? Only 3 kids. There's only 3 examples. If I flip, I know, only is enough. But do you see what I'm saying? I can flip a coin 3 times and get heads every time. That doesn't mean it's a one-sided coin. You see where we're going with that? Because humans don't have tons and tons of offspring, like fruit flies. If you had hundreds of offspring, chances are, if you were a heterozygous individual, you're mating between your husband, you're going to see some of those kids that are recessive, even though he's dominant. Widow's peak. What's that? Yeah, it's the way your hair. You can have a straight hairline or one that kind of points down. So a widow's peak is dominant, huh? Like Dracula, exactly. You're good at Halloween.
The ear thing is fascinating.
Yeah. You never noticed, did you? Who's got attachy earlobes?
Mine are attached.
Yeah, yours are attached. Some of them are hanging free. Mine are free.
Mine are attached.
Yeah, go right straight to your...
Yeah.
So free earlobes versus attached earlobes. Free earlobes are dominant traits. So what you're going to see, and you'll do these little experiments in lab, is it doesn't necessarily, just because a trait is dominant, you don't necessarily see it predominant in the population. It depends on who you what? It depends on who you mate with, exactly. So looking at these family pedigrees can give you some ideas of what genotypes are as we go. Say I have in my family, and you're a perfect example because you did a test cross for us, your husband has freckles. You don't. Your children are a combination. Your daughter, who has freckles, is what? Just judging by looking at your family tree. If we took her and put her by herself, we'd have no idea what she was, her genotype was. But then when we look at you, her parents, and we see what you look like, we know that your daughter is what? Your daughter is heterozygous, right? So family pedigrees can help to fill in the blanks with respect to what genes you carry, just looking at different traits. So dominant traits are not necessarily normal or more common. It just happens to be whatever you happen to see mating in a population. And populations tend to be groups of people in a certain area or groups of organisms in a certain area. There's different traits. Typically wild type traits are those that are seen more often in a group, in a population, in nature. And they're not necessarily specified by dominant alleles. How many fingers do you have? 5. You have 4 and a thumb, which is a finger. Do you know that you can have 6 fingers? Did you know that that was a dominant trait? It is.
It is.
Yep. Have you ever seen anybody with 6 fingers? Nope. It's a dominant trait, though. It should be. We should see it all over the place, right? Why don't you think you see people with 6 fingers? Well, why don't you see, like,
Evolution necessary to do
it's not convenient. How about, let's give an example of difference in the old days. Like, say, around the Salem witch era. If you were born with something different, they'd probably kill you. Yes, because you were what? You were different. If you didn't live long enough to do what? Reproduce, you wouldn't pass that on to your offspring, would you? See where I'm going with that? So just because it's dominant doesn't mean it's better. And just because it's dominant doesn't mean it's going to be more common.
So you're saying that gene's erased out of the human genetic?
It's not, there's probably populations somewhere on this planet that has six-fingered individuals. I don't know. So they're not necessarily specified by dominant alleles. Family pedigrees are going to show the history of traits in a family. And you can analyze human traits based on family pedigrees. What do we have now? I mean, this is how we figured it out way back when. But now what do we have? And we have genetic testing now. But way back when, this is how we'd figure it out. You know, where did that person, Kevin, get his Attache earlobe from? Well, Aaron and Betty up there, what were they? They had free earlobes. But free earlobes is a what? It's a dominant trait. So they both had free earlobes, right? And then they had some kids with what? They had some Attache earlobe kids. How come? Because since they were both heterozygous, some of their offspring were what? Homozygous, recessive. Okay? And then these two guys, Hal and Ina, had their offspring based on the genotypes we figured out from their parents. So Ina was a free female, right? And we didn't know what Ina was until she what? Mated with Hal. And they had some kids, Kevin, who had Attache earlobes. Yes? So that's how family pedigrees can help us with genetics. And then we see in families so many different traits. So you could look at this family and say, okay, are those parents his parents? Or are those parents her parents?
Definitely his.
How can you say? Well,
His mother has the exact same nose as his.
yeah, exactly. Well, the nose, I don't know. He's got his father's nose, but look at her eyes. He's got his mother's eyes. Yes? He's bald, so we're not going to talk about hairline there.
Well,
And then,
he's probably bald because he doesn't want his hair to look like his father.
so he shaves his head.
Which is what my dad does.
Yeah. But you see, in any given family, there's so many things, and you could analyze so many different things. Does it say in the book who's who? It doesn't really say, but I'm calling that's their son, and the grandchildren. Look at the grandchildren. Who do they take after more? Their mother. Yes. So, traits. Oh shoot. So human traits show simple inheritance patterns and are controlled by single genes on autosomes. What's an autosome? We had 23 pairs of chromosomes, right? You have 22 autosomes.
Oh.
And
Read that question wrong on the quiz.
All the ones that are
Your body.
correct. So autosomes. So human disorders controlled by a single gene tend to be, not always, controlled by a single gene on autosomes. And these are some of the different ones we see. You can see this in your textbook on page 155, table 9.1. Lack of pigment in skin, hair, and eyes, albinism, is a recessive disorder. Cystic fibrosis, PKU, sickle cell disease, Tay-Sachs disease. They're all genetically inherited disorders. And these are some on the autosomes in humans. Those are recessive disorders. So in order to have them, what does that mean? You have to have genetically both recessive genes. What if I have a dominant and a recessive gene for that particular trait? Am I going to have Tay-Sachs disease or sickle cell disease or cystic fibrosis? No. But I'm also going to carry those and possibly give them to my offspring. What do they call me? They call you a carrier. Exactly. Dominant disorders. So I only need one of these. Dwarfism is dominant. Alzheimer's disease. Huntington's disease. High cholesterol. I have a hard time saying that word. No, that's why. I'm pushing on the wrong computer. So most human genetic disorders happen to be recessive. Most. Which is kind of good. Individuals who have the recessive allele but appear normal are said to be what? Carriers for the disorder. So this is a hearing disorder. Deafness. It's a recessive disorder. If I have two parents who are what? Heterozygous for this disorder, and they mate, the chances that they have a deaf offspring are going to be what? 1 in 4. The other 3 are going to be hearing. Correct. So 3-to-1 phenotype for this. Yes? But it's 1-to-2-to-1 for genotype. So 1 dominant, 2 heterozygous, and 1 recessive. So cystic fibrosis, most common lethal genetic disease in the United States, caused by a recessive allele carried by about 1 in every 31 Americans. Again, because it's recessive, we don't tend to see it that high in the population, unless two what? Heterozygous individuals, carriers, cross, and their offspring happen to be recessive for the trait. Prolonged geographic isolation of certain populations can lead to what we call inbreeding. So when I breed too closely, if I take a heterozygous individual and breed them with a homozygous dominant individual, what are all my offspring going to be? There's only, not heterozygous.
What was the question?
They're all going, if I take a heterozygous individual for the trait, so let's call it, let's use F, because we can see the letters better. Yes? What's my Punnett square going to look like?
Everyone's gonna carry the tree.
No. See? It's going to be a 50-50. So half of my offspring could be carriers. Are any of them going to have the disease? Whatever the disease I happen to be talking about is? No. So we're going to have 2 carriers. They're all going to be normal. But if I start inbreeding, yes? What am I going to start to see more in my offspring? Yeah, exactly. And if I inbreed even closer, maybe these guys, yes, I start to narrow down my gene pool. That's why it's important for us as humans to mix it up. Yes? Again, that's why you're not supposed to what? No marrying your sister or your first cousin. So inbreeding increases the chances of offspring that are homozygous for harmful recessive traits. That's why that's a law. Because we tend to enhance harmful recessive traits when we breed too closely. Some human genetic disorders are dominant. Achondroplasia is a form of dwarfism. That is a dominant trait. So how come everybody in our population, or the majority of the population, aren't dwarfs or short in stature? Huh? No, not necessarily.
Well, because of the dying, the embryo dying.
Yeah, so homozygous dominant genotype causes death. So we're never going to see anybody in the population that has this. Because that will, what will happen to that during embryonic development? It'll die. So what do we see in our population? We see heterozygous individuals. So heterozygous individuals are going to have the disorder. So heterozygous individuals can only mate, yes? Because they're the only ones that stay alive to mate if they're dwarfs. What are the kids going to look like? It's dying. Dead. Yes? Dwarf. Dwarf. Normal. Normal stature. Yes? So, there's only a 3 to choose from with this particular trait, because this one won't survive to reproduce. Huntington's disease, which leads to a degeneration of the nervous system, does not usually begin until what? Middle age. So you can, oh, there's my short in stature individual. And did you ever watch this show? I guess it was a reality show.
I've seen it before.
What was it called? Yeah. So the parents are here, Matt and Amy. 3 of their kids, normal stature. 1 of their children has the dwarfism gene. So that doesn't follow the rules at all, right? But remember, every time the mating goes on, what is the chance that we're going to have normal stature? 1 in, no, not 4. 1 in 3. Yes? Because that one's not going to make it. Yeah, it's still 1 in 4. It's still 1 in 4 probability. But in this case, 3 out of their 4 children are normal in stature. Yes? So, observation, pooches. Dogs come in a very wide variety of physical types. Where do all dogs come from genetically? Wolves. What's the basic genetic basis for canine coats? So composition of genes of a wide variety of dogs with different coats would identify the genes responsible. Mutations in just a few genes account for coat appearance. Mutations, isn't that bad? Mutation accounts for change. Not always bad, sometimes good. Compared the DNA sequences of 622 dogs from dozens of breeds. 3 genes in different combinations produce 7 different coat appearances, from very short hair to full, thick, and wired hair. So we know that 3 different genes in the dog, canine, are responsible for coat. And the combinations of these genes give us all the different coats that we see. Today, many tests can detect the presence of disease-causing alleles. We can map out genes because we've done something called gene mapping to figure out on where these loci appear, on what pair of genes they appear on, or pair of chromosomes. Most genetic tests are performed during pregnancy. Has anybody ever had these tests done? No? You guys are a little on the young side. Those of us older ones, they never did that stuff for us. So amniocentesis, we actually look at cells that have sloughed off the child in the amniotic fluid. And then we can actually take a little sample of the tissue that child is helping them grow, called the chorionic villi, and we can remove cells there to look at the map, that karyotype. If certain things run in your family, if there's concerns about the offspring's genetics, you can use this to test and see what the genetics of your offspring are going to be. Some patterns of genetic inheritance are not explained by Mendel's law. So there's variations. Mendel didn't hit every single thing, right? Because he was just working with what? Peas. He got a lot done, but there are other things that take into account the appearance that we see. We worked a little further to come up with something called incomplete dominance. So F1 hybrids have an appearance that's sort of between the phenotypes of 2 parents. And they're not a mix of, they're kind of between. Now, Christmas is coming up, right? So I can give you an example of this. Do you ever try to make a red cookie? Those of you bakers out there. How do you make a red cookie? You use food coloring, right? But in order for it to be red, it seems like you have to use a ton of food coloring, right? Think of that example when we talk about the law of incomplete dominance. It's not that the trait blends together and disappears to become something else. It's just that if I only have one shot of it, the appearance is not going to be what I want it to be, just like my food coloring. So pretend red flowers have 2 shots, 2 big huge squeezes of my red food coloring. So they look red. If I mate them with white, and this isn't really a dominant recessive kind of thing, I get an intermediate guy trying to make those cookies with only 1 shot of food coloring rather than 2 shots. You with me? So this intermediate, this F1 generation, this pink flower, only has 1 copy of that genetic material that's producing that red color. The other copy is producing what? White. And when I mix white and red, what do I get? Pink. We know that it doesn't go away because some people thought or called it the blending theory. And once they blended together, those colors would never come back again. Why do we know that's not the case? Why do we call it independent? Yeah, because I can see them again. So if I take my pink flower, and we're going to use those as our genotype, red and white, and I cross them, what do I end up getting? Oh, damn. I get a red again. Red comes back. Yes? I get two pinks. And I get a white. So those original colors come back in my offspring.
Did that kind of explain it by color?
Not really. It's a mutation of the gene, so we get variations in colors.
It's like green eyes.
Yeah, green, blue, hazel. Those are all different. Yeah, and sometimes the way each of your eyes express their genetic material, sometimes you can have two separate colors. Some people have two different colored eyes. So your environment is also going to determine how your genes are expressed. But this is the best example I can use is the red cookies at Christmas.
Mendel must have, when we're going with the purple and the white, he must have experienced that in a different shade of.
No, because it's a dominant recessive trait. If he had looked at a different flower that expressed their genetics in a different way, because we look at all the variations of flowers that we see nowadays. Sometimes we see this. Sometimes we see flowers with white and color. So all these different genetics, genes express themselves in different ways in different species. And we see many combinations of that now. But he was just working with those pea plants. This is a snapdragon, I think it's called. So we see a different type of expression of those genes. Again, we call this incomplete dominance. We're going to see something different in the offspring, pink, but it doesn't totally mask the recessive trait. Yes? And it has to do with the fact, just like making my cookies, if I had 2 shots of red food coloring, 2 copies of that red gene, I would see a red flower. If I only have 1 copy of the red gene, I'm going to get that intermediate pink because I only have 1 shot of food coloring in my batter. It's not as bright. Make sense? So it's like mixing red and white. Hmm?
It did up until now.
Oh, why? Did I make it confusing?
No, I guess I'm just not understanding why.
But again, think of the food coloring, that pink flower, just like my cookies. There's 1 shot of red food coloring and 1 shot of white food coloring. 1 gene for red, 1 gene for white. They're both expressing themselves and making proteins. One of the proteins is red, one of the proteins is white. In this particular trait, when you mix red and white together, what do you get? Pink. It's not like the brown and blue, where brown actually masks the blue, even though they're both there. And that's happening in brown and blue, too. The brown is masking the blue. In this case, they're kind of working with each other. And again, that's incomplete dominance. We see that in plants and people. Cholesterol. Incomplete dominance in plants and people. Who has a high cholesterol? Anybody? No, I never did until I got older. Believe it or not, well, you know when I started to develop a high cholesterol? When I lost weight and started to exercise. Whose fault is it, though? Yeah, it's my father's fault. I always blame him. So it's a human trait that's an example of incomplete dominance and characterized by dangerously high levels of cholesterol in blood. So some of us have high cholesterol because of our genetics, not necessarily because of what we eat. Heterozygotes have blood cholesterol levels about twice normal. Homozygotes have about 5 times the normal amount of blood cholesterol and may have heart attacks as early as age 2, depending on how severe it is. So what would you rather be? Yeah, if I had to be a heterozygous, which is probably what I am, because I didn't show up till later for this trait. And it has to do with our different cholesterol carriers and the receptors on our cells. So homozygous for the ability to make a little receptor called LDL. If you're homozygous for that, you have normal cholesterol levels because LDL is going to take care of your cholesterol, pass it around. Actually, it's a precursor to something called HDL, which is the guy that's going to take care of your cholesterol levels for you. If you're a heterozygote, you don't have as many of those receptors on your cells. So you have a little bit of a high cholesterol. If you're homozygous inability to make those receptors, period, cholesterol is going to be very high in you because most of it's going to be floating around in your circulatory system rather than hanging out on the cells where it should. So we see varying degrees of that particular disease. Not to make anything even more confusing, but now we're talking about something called multiple alleles and codominance. So black or brown, red or white. Sometimes there's more than 1 or 2 different combinations that you can have. When we talk about your blood type in humans, the ABO blood group is an example of multiple alleles. So, you have proteins on the surface of your cells. They're called antigens. And what codes for the making of any given protein? Your genetics, your DNA. So those proteins on the surface of your cell has some sort of genetic information that codes for it, yes? If a donor cell has a carbohydrate, and it's actually a protein with a carbohydrate on it, that's called A, or B, you have them on the surface of your cells. Has anybody ever done their blood type? Donated blood? Do you know what's your blood type? You're B positive. You're A negative. I'm A positive. There's 3 different ones you can have on the surface of your cells. You can have A, or you can have B, or you can have neither of those. And if you don't have either of those, what do we call you? We call you O. These guys are called antigens. And they can cause other organisms to produce protective chemicals against them. Those protective chemicals are called antibodies. And you do this all the time. When you come in contact with a foreign substance, say that was a virus with little proteins or substances on the surface of their cells, and they came into your body and you recognized them as foreign, what would you do? And you'd fight them off. And one of the ways you'd fight them off is to make a substance that would sort of neutralize them. And that substance is called a what? It's called an antibody. So the immune system produces these proteins called antibodies that can bind to these proteins with little carbohydrates on them that give us our blood type. Oops. Keep hitting the wrong screen. So, who had type B blood? Okay. If I give you my cells, I'm a donor. Blood cells have type A on them, and I give them to you. Your body's going to recognize them as what? They're foreign to you. And the blood cells are going to clump in your body because you make a substance to protect you against any foreign ABO blood grouping that comes in. It's called an antibody.
So what are antibodies?
They're substances that neutralize antigens.
So
They're
it's
proteins.
cells of the cell.
Not to confuse you, but it can be on the surface of the cell, but typically it's floating around in the circulatory system.
Substance, not an actual
It's a
cell.
protein.
Okay.
Yes. So, if I give type B my type A cells, these are little squares. That person is going to make these little antibodies that float around in their circulatory system. And this is probably one of the worst ways to make you understand this because it's confusing. I wish they didn't use this example in your book. These antibodies that are floating around in type B's body that they made against this antigen are going to attack my cells. And you know what's going to happen? Big clumps. If there's big clumps of cells floating around in your circulatory system, is that going to work very well? Nah, no. That'll kill you. Yes? Yep, kind of. Except it's not really clotting, it's clumping. There is a difference, believe it or not. Let me give you a better example of antigens and antibodies. This is a weird thing that happens in humans. We typically don't make antibodies against a foreign substance unless the foreign substance comes into our bodies. So chickenpox, for example. You don't have antibodies to chickenpox unless what? You get the chickenpox.
What about the vaccine?
What does the vaccine do? It does, yeah, it gives you something. Viruses have their own little...
They take all the DNA out of it and like just delete the shell so your body can recognize it.
Correct. Viruses have their own little signature. They have their own little antigens on the surface of them. Your body recognizes them as foreign. Your body makes antibodies. We kill off the virus. Correct. Yeah, that happens. It's not always 100%. And it has to do with your body's ability to make enough cells to make enough antibodies. That's why it's really important when you get a vaccine that you have to follow up with what? Yeah, either boosters or they have to test and see how much antibody you have in your system. Because sometimes your immune system doesn't kick in enough. So if you don't have enough, you have to get another booster. And some antigens need boosters normally, like mumps, measles, rubella. That needs a booster. So, blood types. 3 different kinds. Remember, you have 2 genes, right, for blood type. We call that gene I. So you can either have IA, or you can have IB, or you can have neither one of those. We call you little I. Watch. This is where the codominant stuff comes in. So what if this is my genotype? What do my cells look like?
Both A and B receptors.
I have both A and B. A and B proteins on the surface of your cells.
Now O will work with like any, right?
And what do O have? Not really nothing. They just don't have any A, and they just don't have any B. So this is AB, this is O, and what's type A look like? Just A. And what's the other type I can be? I can be. Yes?
So we'll say I was a type AB. I can get blood from a type B person or a type A person.
Correct.
Or a type O.
But you can only get their cells,
Not their blood.
not their whole blood. Because, and this is the weirdness of the whole blood typing thing. Exactly. Your body, and this is the only, and we don't even know why this happens. We still don't know why this happens. But you make antibodies against the antigen that you don't have. So does type AB make any antibodies? No. Against A or B? And we're just talking A and B. How about type B? What type of antibodies will they have floating around in their blood? Anti-A antibodies. How about type A? Anti-B antibodies. How about that guy? Both. Yeah.
Oh yeah, that's the universal donor.
Only what? Cells. No plasma. Your blood is 2 parts. Yeah. When you go and you donate blood, you know what they do to your blood? They separate it, exactly. So they're going to take your cells, red blood cells, wash them off really, really good, and put them in a bag with the perfect concentration of saline. When you get blood at the hospital, you're only getting cells. You don't get whole blood. Yeah. That red blood cells. That's what makes it red.
So what do they do with your plasma?
They separate it and they can give you just plasma too.
Why?
Or they pull out some different important factors in your plasma that you could give to patients. For example, if someone has hemophilia, they have certain chemicals within your blood that they need because they're lacking them. They can pull those out of your plasma and give them to the patient. They don't need all the other stuff. So that's what they typically do with your plasma. That's a different, yeah, negative and positive, that's a different protein on the surface of your cell. It's called RH or D. So RH positive would also have these guys. So this would be AB positive. This would be O positive. If I didn't have any D's, what would I be? Negative. Mrs. B, negative. Okay?
Do we do blood typing in our labs?
You should, yeah? They have some samples you can do. It depends on your instructor. But okay, so we obviously didn't finish this, did we? So we will finish